Good morning. Today we are going to take the fourth part of our first chapter, Life's Mysteries in Little Chambers. Okay, so this is the last part of our first chapter, and today we are going to study about how to prepare an observation material to view through a microscope. I have already told you that we can't keep an object. The object to be viewed through a microscope is known as the specimen. So we can't directly keep the specimen on the stage. You know about the different parts of the microscope now. So we can't keep the object or the specimen directly on the stage. So we are using, for keeping a specimen, we are using a slide. A slide is the rectangular shaped glass piece. So now you can see, we can see which are the steps needed to prepare an observation material. Okay. So in this, you can see the first step, that is, put the material to be observed in the Petri dish containing water to prevent dehydration. That means if you wanted to see the section of a stem, ഒരു സ്റ്റെമ്മിന്റെ ഇന്റേണൽ എന്തൊക്കെയാണ് ഉള്ളിലുള്ളത് നിങ്ങൾക്ക് കാണാനാണ് ഒരു മൈക്രോസ്കോപ്പ് കൂടെ ഒബ്സർവ് ചെയ്യേണ്ടതെങ്കിൽ ആ സ്റ്റെം, ആ പീസ് ഓഫ് സ്റ്റെം അതല്ലെങ്കിൽ ആ പീസ് ഓഫ് റൂട്ട് യാതൊരു ഒരു ഗ്ലാസ് ഡിഷാണ് പെട്രി ഡിഷ് എന്ന് പറയുന്നത്. അതിൽ വിത്ത് വാട്ടർ, അതിൽ വെള്ളം you have to put the stem or root piece. Is it clear for you? In order to prevent the dehydration. അതിൽ നിന്ന് വാട്ടർ ലോസ്റ്റ് ആവാതിരിക്കാൻ, സ്റ്റെമ്മിൽ നിന്നോ റൂട്ടിൽ നിന്നുള്ള വാട്ടർ ലോസ്റ്റ് ആവാതിരിക്കാൻ വേണ്ടിയിട്ടാണ് പെട്രി ഡിഷിലെ വാട്ടറിൽ നമ്മൾ റൂട്ട് പീസോ അതല്ലെങ്കിൽ സ്റ്റെമ്മിന്റെ പീസോ which material we want to observe, that is we are keeping it in the Petri dish in the water. That's the reason we are keeping the material to be observed in the water in order to prevent the dehydration. Dehydration means the loss of water. Then the second step, take thin cross section of the material to be observed. We are using a blade. Blade use ചെയ്തിട്ട് അതിന്റെ thin ആയിട്ട് കട്ട് ചെയ്യാം, സെക്ഷൻ എടുക്കാം. ആ സെക്ഷൻ എടുക്കുമ്പോൾ ശ്രദ്ധിക്കേണ്ടത് അതിന്റെ അറ്റം അല്ലെങ്കിൽ പെരിഫറി, അതായത് അതിന്റെ എൻഡിങ് പാർട്ട് അല്ലെങ്കിൽ കറക്റ്റ് സർക്കിൾ ഷേപ്പിലുള്ള സെക്ഷൻ എടുക്കണം. അത് വളരെ തിന്നായിരിക്കണം, കട്ടിയിലുള്ള സെക്ഷൻസ് എടുക്കരുത്. അങ്ങനെയാണെങ്കിൽ നമുക്ക് ഒബ്സർവ് ചെയ്യാൻ ഡിഫിക്കൽട്ട് ആയിട്ട് മൈക്രോസ്കോപ്പ് കൂടെ നമുക്ക് ക്ലിയർ ആയിട്ടുള്ള ഒരു പിക്ചർ അതിന് കിട്ടില്ല. സോ ടേക്ക് thin cross section of the material to be observed using a blade. You have to take the thin cross section of the stem or root to be observed. Then with a brush, shift the sections to a watch glass containing water. So we are shifting all the sections, cross sections we have already taken. We are shifting it using a brush, not using the hand. From the blade, we are shifting it using a brush into the watch glass containing water. So watch glass, it's containing water. Into that, we are again shifting using the brush. The cross sections we are shifting to the watch glass. Then stage 4, put the thinnest and complete sections to a watch glass containing stain. Stain എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ ഒരു കളർഫുൾ ലിക്വിഡ് ആണ്. ഡിഫറെന്റ് ടൈപ്സ് ഓഫ് സ്റ്റെയിൻ ഉണ്ട്. അപ്പൊ ആ സ്റ്റെയിനിലേക്ക് നമ്മൾ സാഫ്രാനിക് സ്റ്റെയിൻ ഉണ്ട്, ലീഷ്മാൻ സ്റ്റെയിൻ, അങ്ങനെ ഡിഫറെന്റ് ടൈപ്സ് ഓഫ് സ്റ്റെയിൻസ് ഉണ്ട്. അപ്പൊ ആ സ്റ്റെയിനിലേക്ക് നമ്മൾ ഈ ക്രോസ് സെക്ഷൻസ് തിൻ ആൻഡ് കംപ്ലീറ്റ്, അറ്റം മൂലൊന്നും പൊട്ടിയതായുള്ള ക്രോസ് സെക്ഷൻസ് എടുക്കരുത്. കംപ്ലീറ്റ് ആയിട്ടുള്ളതും തിന്നായിട്ടുള്ള തിക്കായുള്ള സെക്ഷൻസ് എടുക്കാൻ പാടില്ല. സോ പുട്ട് ദ തിന്നസ്റ്റ് ആൻഡ് കംപ്ലീറ്റ് സെക്ഷൻസ് ടു എ വാച്ച് ഗ്ലാസ് കണ്ടെയ്നിങ് സ്റ്റെയിൻ. സ്റ്റെയിൻ ഒരു കളർഡ് ഫ്ലൂയിഡ് ആണ്. ആ ലിക്വിഡിലേക്ക് നമ്മൾ എന്ത് ചെയ്യുന്നത്, ഇത് ട്രാൻസ്ഫർ ചെയ്യുന്നത്. എന്തിനാണ് നമ്മൾ സ്റ്റെയിൻ ചെയ്യുന്നത്? ഫോർ വാട്ട് പർപ്പസ് വി ആർ സ്റ്റെയിനിങ് ദ material? Because in order to see the section clearly through the microscope, in order to see the parts or the cells clearly through the microscope, we are using the stain or staining the material to be observed through the microscope. Then next, stage 5. Here we are taking the slide. I have told you a rectangular shape glass piece is the slide. On that slide, we are putting, we are keeping a drop on the slide. We are keeping a drop of glycerin. For what purpose we are keeping the drop of glycerin on the slide, in the center of the slide. For what purpose? In order to avoid the drying up of the material. So if we are simply keeping the material on the slide, what will happen? It will dry. The water from the material will be lost. So in order to prevent that, in order to prevent the drying of the material, we are keeping a drop of glycerin in the center of the slide. Then stage 6. What is happening? Wash the stained section in clean water and place in the glycerin on the slide. For what purpose we are again washing the stained specimen? Stain ചെയ്ത സ്പെസിമെൻ ഒന്നും കൂടി വാഷ് ചെയ്യുന്നതെന്ന് വെച്ചാൽ എക്സ്ട്രാ സ്റ്റെയിൻ അതിലുണ്ടെങ്കിൽ അത് പോകാൻ വേണ്ടിയിട്ടാണ്. എക്സ്ട്രാ സ്റ്റെയിൻ ആയിട്ടുണ്ടെങ്കിൽ നമുക്ക് എന്താണ് ഒരുപാട് കളർഫുൾ ആയി കഴിഞ്ഞ് കഴിഞ്ഞാൽ നമുക്കത് കാണാൻ ബുദ്ധിമുട്ടായിരിക്കും. അതുകൊണ്ടാണ് എക്സ്ട്രാ സ്റ്റെയിൻ റിമൂവ് ചെയ്യാൻ വേണ്ടിയിട്ടാണ് നമ്മൾ പെർഫെക്റ്റ് ആയിട്ടുള്ള ഒരു ക്രോസ് സെക്ഷൻ എന്ത് ചെയ്യുന്നത്, തിന്നസ്റ്റും പെർഫെക്റ്റ് ആയിട്ടുള്ള എവിടെയും മുറിഞ്ഞു പോവാത്ത അല്ലെങ്കിൽ ബ്രേക്കപ്പ് ചെയ്യാത്ത തിന്നായിട്ടുള്ള clear cross section നമ്മൾ എന്ത് ചെയ്യുന്നു, സ്റ്റെയിൻ ചെയ്തത് നമ്മൾ വീണ്ടും വാഷ് ചെയ്തിട്ടുണ്ട് in order to remove the extra stain. എന്നിട്ട് അത് we have already kept a drop of glycerin on the center of the slide. So on that glycerin we are keeping that perfect cross section of the specimen. So we are keeping it. Why we are keeping a drop of glycerin in the center of the slide? In order to avoid the drying up of the material. Is it clear for you? And another thing is why we are washing the already stained material, in order to remove the excess stain from the material to be observed or specimen to be observed. Then after that, place a cover glass over the material. Cover glass is a square shaped thin glass material. Okay, so place a cover glass over the material using a needle in such a way that air bubbles do not pass. അപ്പോൾ അതൊരു നീഡിൽ ഉപയോഗിച്ചിട്ട് മെല്ലെ മെല്ലെ വേണം ഡ്രോപ്പ് ചെയ്യാൻ വേണ്ടിയിട്ട്. പെട്ടെന്ന് കവർ ഗ്ലാസ് അതിന്റെ മുകളിൽ ഇട്ട് കഴിഞ്ഞാൽ സ്പെസിമെന്റെ മുകളിൽ ഇട്ട് കഴിഞ്ഞാൽ എയർ ബബിൾസ് എൻ്റർ ചെയ്യും. ഇഫ് എയർ ബബിൾസ് ഇസ് എൻ്റർഡ് വി ആർ നോട്ട് ഏബിൾ ടു സീ ദി സ്പെസിമെൻ ക്ലിയർലി. നമുക്ക് മൈക്രോസ്കോപ്പ് കൂടെ ഒബ്സർവ് ചെയ്യുമ്പോൾ അത് എയർ ബബിൾസ് എൻ്റർ ചെയ്ത ഒരു സ്പെസിമെനെ ആണെങ്കിൽ, ഒരു സ്ലൈഡ് ആണെങ്കിൽ നമുക്കത് ക്ലിയർ ആയിട്ട് ആ പിക്ചർ കാണാൻ, ക്ലിയർ ആയിട്ട് അതിന്റെ പാർട്സ് കാണാൻ ഡിഫിക്കൽട്ട് ആയിരിക്കും. സോ ദാറ്റ് വി ആർ യൂസിങ് എ നീഡിൽ ആൻഡ് keeping the cover glass slowly on that specimen in order to avoid the air bubbles. Then last stage, so we have prepared a slide. Now the slide is ready, and the last part is that wipe out the excess glycerin using tissue paper. So we have to clean that remaining part of the slide, that is talking about the excess glycerin that is spread over the remaining part of the slide. We are using a tissue paper for that and cleaning. After that, we are keeping that slide on the stage of the microscope and using the stage clip. We are holding it on the microscope, and by adjusting the knob, using the adjusting knobs, we are fine adjustment and coarse adjustment knobs, we are adjusting the distance between the specimen and the objective lens, and we are able to view the object or specimen through the microscope. Is it clear for you? So I think the steps for doing how to prepare an observation material is clear for you. One thing you have to remember is, that is, for what purpose we are keeping the specimen in the water in the petri dish. The question comes like that, in order to avoid the dehydration of the material. Dehydration means losing of the water, that is losing of the water from the material. So in order to avoid the dehydration, we are keeping the material in the petri dish in the containing water, petri dish containing water. Is it clear for you? And for what we are using the glycerin? We are using the glycerin in order to avoid the drying up of the drying up of the material that we are going to observe. Then the third thing, that is for what we are washing the stained material. Again, washing the stained material with water in order to remove the excess stain in the material to be observed. Is it clear for you? And these are the things you have to keep in mind, and you have to study the steps. And you have to study the steps in the order or all steps we are doing to view a object to be observed or a specimen to be observed through the microscope. We all know that we can't directly keep that object or specimen on the stage of the microscope. We have to prepare a slide for that, and we can keep it on the stage and we can view through the microscope with the help of the eyepiece lens we are viewing, and with the help of the objective lens, it is magnifying the object. Is it clear for you? Now we are going to study about the next one of the cell organelle, that is plastids. Is it clear for you? Plastids are the specialized cell organelle that is seen only in the which cell? Plant cell only. Plastids are specialized cell organelles that is seen only in the plant cell. And what's the peculiarity of the plastids? Plastids are double membrane, having a double membrane, and there are three kinds of plastids. They are chloroplast, chromoplast, and leukoplast. Chloroplast, it consists of, it helps in the, it consists of the chlorophyll pigment. Chloroplast consists of the chlorophyll pigment, and it helps in the photosynthesis. Then chromoplast, it consists of the pigments, that is, the name of the pigment is xanthophyll, that is yellow in color, and the carotene, that is yellowish orange color, and anthocyanin, that is purple or red color. These pigments, and these pigments are providing the color to the fruits and leaves. You are seeing, apart from the green color, you are seeing different colors of flowers and leaves and fruits. All these are seen because of the presence of the chromoplast. Then next is the leukoplast. Leukoplast is the colorless plastid. And Shifa, what you are doing there? Are you seeing watching the class or playing with the? So those who are watching the video, sit properly and view the class. Otherwise, I will call you after the class. So what we are telling about the leukoplast. So leukoplast is the colorless plastid. Colorless plastid, it is stored in the food, where the food materials are stored. This leukoplast is seen in abundance. Then another thing is, there comes a question: how the color and taste of the fruits are changing when it is ripened? So that the answer for that is the chloroplast present in the fruit is changed into chromoplast when it is ripened, and the starch present in the fruit is converted into sugar. That is, it is sweeter when the fruit is ripe. That is the reason behind why the fruits appear to be ripe. Now I will show you a video regarding the plastid that will make you more clear about the plastid, different kinds of plastid and functions. Everything will be more clear from that.
Now that the entire city was almost built, people started living in the city. So a permanent source of food was now required to sustain the entire population. So the mayor set up a number of canteens in various parts of the city which provided the people with food. Similarly, plants have such canteen-like structures which manufacture food, and these canteen-like structures are known as chloroplast. Chloro means green and plast means who form. So what gives the chloroplast this green color? Well, there is a pigment present inside the chloroplast which is known as the chlorophyll. This chlorophyll is green in color, and that is why even the chloroplasts are green colored. These chloroplasts are found in abundance in green leaves. The chlorophyll present inside the chloroplast absorb sunlight and together with carbon dioxide and water manufacture food by the process of photosynthesis. So, do the leaf cells carry only chloroplasts? Let us see. There are a large number of cells in the leaf. Now let us focus on one cell. Yes, you see abundant of chloroplasts present in the leaf cell. But what are these white structures and these colorful red structures present in the leaf cell? Well, these are known as the chromoplasts and the leukoplasts. The white structures are known as the leukoplasts. The red colorful structures are known as chromoplasts. Now let us study further about the leukoplasts. Your mother cooks food for you every morning. Now what if she cooks some extra one day? Will she throw away the extra food? No, she won't. She'll store the excess food in the refrigerator for future use. Similarly, all plant cells manufacture food by the process of photosynthesis. Now, the extra food that is present in the plant gets stored in organelles known as the leukoplasts. Leuko means white or colorless. So, leukoplasts are organelles present in the plant cells that store these excess food. Potatoes, something that you all love. Now, can you tell me why potato slices have a white appearance? Well, that is because potatoes have an abundance of leukoplasts that store starch in them. Now, you can carry a simple experiment. Just take a potato slice, stain it with iodine, which is specific for the starch granules present inside the leukoplast, and observe it under the microscope. This is what you see. You see purple granules of starch, and this proves that potatoes have an abundance of leukoplasts in their cells. Now, have you seen an unripe tomato on the plant? Well, if you have, you'll see that they are green in color. Very different from the red tomatoes that you have in your salads. Now, why does the color of the tomato change from green to red? This is because of the progressive decrease in the number of chloroplasts and the progressive increase in the number of chromoplasts. So the green color decreases and gives way to the more prominent red color. So what are these chromoplasts? Well, chromoplasts are organelles present in the plant cells that have many bright pigments inside them. So chromoplasts are organelles that give the plant different colors except green. Now see, if you see a rose cell under the microscope, you will find a lot of chromoplasts carrying a bright red pigment. And this pigment is giving the rose this bright red color. So the two most common pigments that are present inside the chromoplasts are xanthophyll and carotene. Xanthophyll is responsible for the yellow color of bell peppers, and carotene gives the bright orange color in carrots. So we've talked about the chloroplasts, the leukoplasts, and the chromoplasts. Now notice that all these three structures are double membraned. See, they all have double membranes. Now all these three structures can collectively be called as plastids. So plastids consist of chloroplasts, leukoplasts, and chromoplasts. Now, if you've been paying attention, you will realize that I have just talked about plastids in a plant cell. That is because plastids are present only in the plant cell. Animal cells do not have plastids. This means that animal cells do not have chloroplasts in them, so they cannot manufacture food by the process of photosynthesis. That is why we are dependent on plants for food. All animals and all human beings depend on plants for food. Now you can tell me that carnivorous animals are not dependent on plants for food. They have other small herbivorous animals. No, even carnivorous animals are indirectly dependent on plants for food. How? They have small herbivorous animals, and these herbivorous animals are dependent on plants for food. So indirectly, even carnivorous animals or human beings who have only flesh and meat are also indirectly dependent on plants for food.
Or the animal cell. In animal cell, you can see, I will show you the picture of the animal cell. Okay, in that I will explain it to you. And the picture of the animal cell, it's having a cell membrane, and here the cell wall is absent. You have seen in the plant cell wall is present, the animal cell wall is absent. Then another thing, you can't see chloroplast in the animal cell. So chloroplast is also absent in the animal cell. Another thing is that ribosome, cytoplasm, mitochondrion, endoplasmic reticulum, Golgi complex, nucleus. All these you can see in the plant cell also. The two cell organelles that is not seen in the plant cell, that is centrosome and the lysosome. So first is the centrosome. Centrosome is the cell organelle that is seen only in the animal cell, and the function of the centrosome. Centrosome consists of the centrioles, which helps in the cell division. And the next is the lysosome. Lysosome is also another cell organelle that is seen only in the animal cell, and it consists of the digestive enzymes. These digestive enzymes distract the foreign materials that is entering the cell. So these are the two cell organelles, that is centrosome and the lysosome. Once more I will tell, centrosome consists of centrioles, which helps in the cell division. It is only seen in the animal cell, and lysosome is the another cell organelle which is seen only in the animal cell, which helps in the which consists of digestive enzymes, which helps in the destruction of the foreign materials that is entering the cell. Now last we can see the difference between the plant cell and the animal cell. We have already told that animal cell do not have cell wall. Plant cell have a cell wall made up of cellulose. Then animal cell do not have chloroplast. Plant cell, they contain chloroplast. Then as they have animal cell, they have centrosome is present. For plant cell, centrosome is absent. They do not have centrosome. Then animal cell vacuoles are small in size, and in plant cell vacuoles are larger in size. And in animal cell, maybe vacuole is not seen. Sometimes vacuole is present also, it is in smaller size in the animal cell. Then in animal cell, lysosomes are large in number, and plant cell lysosomes are absent, and it's present very few in number. And almost in plant cell, lysosomes are absent. Then animal cell prominent Golgi bodies are present, and plant cell subunits of Golgi bodies known as dictyons are present. So I think it's clear for you about the difference between the plant cell and animal cell. So our chapter is completed. Notes I will give you through the WhatsApp, and if you are having any doubt regarding the class, you can ask me through the WhatsApp. Okay, thank you.